So, okay, unfortunately, we just have this kind of microphone. I will circle it around, but first, perhaps I start with the first question. My first question would be to all of you, what were the major steps in the last 5 years in your field? And what is the most challenging new thing you think about now for your research part? Perhaps I start with Johannes and then asking for identification, perhaps.
So my field is identification?
Somehow, yeah, message identification, goal-oriented communication. So
I'm happy I don't have to answer for all of information theory. Yeah, I think an identification, if I'm right, then in the last 5 or slightly more years, actually the whole thing got a lot more traction than before. So kind of it was somewhat unearthed, or it was not completely in the earth, but I think the interest has grown a lot because in general, I think there was more attention towards things like goal-oriented communication and getting more efficient. So since we have very efficient codes to achieve capacity, then the question, if you want to be more efficient, is, well, maybe you have to tailor your model more towards or your coding more towards the actual problem that you want to solve, and not only solve a general problem to mediate all kinds of problems. And I think maybe major steps, I think, have been really the development of the theory of deterministic identification. I think there hasn't been a lot to this before it has been started again. And also, I think just the whole theory with regard to robustness, to, for example, jamming and security and some multi-user problems was very much extended. I think there is still also a lot to do. So I don't think really we have a comprehensive theory of identification in all sorts of situations. I think that's really, there's still a whole way to go. And also, maybe you remember this this picture with all the different scalings. This is something completely new. I think we knew of the exponential thing and of the doubly exponential thing, but all the rest is from the last 5 years.
Okay, so I started working on molecular communication when I started my PhD in Georgia Tech many years ago. No, not so many, but quite. And then, we were putting together the first communication theory aspects regarding substituting the electromagnetic waves with molecule diffusion. And so how does the diffusion channel work? How it is transmitted, received, what is the capacity in diffusion-based systems when free diffusion is there. And then when I started starting being a faculty in Nebraska, I wanted to see the systems a little bit more on a practical viewpoint. And so I was researching what is the best technology that I can use to build these systems. And the most promising, in my opinion, was synthetic biology. So I started to study biology and synthetic biology without becoming a biologist. I was talking with Olaf right now about this, the important, the power of being interdisciplinary and having ideas is inversely proportional to the level of detail that you want to get to on other subjects. And so sometimes it's very valuable to understand the subject, like synthetic biology for me. But if I were to go over all details, I would need another degree, which would require a lifetime. So we cannot do that, right? So my attention was understanding synthetic biology and understand what I can do with the tools that are already in place and what I can understand from a very 1,000 feet overview. And then starting from there, I started studying molecular communication with simple organisms. So I look at bacteria that can be engineered and for which we can have fairly good models that are quite analytical in their shape. And then I move more and more towards understanding how information is processed by these organisms, and I will talk more about that tomorrow. But that sparked my interest in information theory applied to biological systems, simple ones first. And then I evolved my thought, thinking, well, okay, for these simple organisms, I can have fairly good models, and still I can understand there is something that Shannon theory, and I want to go to the theme of this, right? So yeah, Shannon theory does not cover, right? And we came up with this concept of subjective information, and it's just the tip of the iceberg. Because subjective information means that that the organisms attach a certain value to information that we usually don't see in our communication systems that we engineer, right? The electrical ones. Because organisms use information for a goal. And the goal can be very easy or very complex. And so this interpreting information through the subjective information lens can be easy and complex at the same time. And so then we get to these days. So I started being interested in more complex organisms. And so I started looking at the brain. And that's why very interesting for me. And this is something that I haven't really published yet, but it's ongoing. And it's studying how humans interpret information and give value to information. And it's a whole other layer of models. And for that, I had to abandon the analytical models because the brain is a black box. So far, okay? I hope it will not be in 50 years or in 100, but now it's a black box. It has many black box components that we don't know, and we don't know how they work. And so what we use to interpret then is artificial intelligence, so machine learning, actually. And so now my latest research is on machine learning applied to intelligence and how intelligence processes information. So that's my mental trip in a nutshell. Thank you.
All right, so in terms of DNA-based data storage, I would say that the major roadblock right now is synthesis cost. If you remember the figure, I think like to synthesize a megabyte of data, you need $4.5K, not cheap. So I think one of the major breakthroughs in this field was composite DNA, because people realize that, okay, because there is so much redundancy when you're synthesizing strands on an array, because if you want to synthesize A, C, 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 G, G, T, whatever, you're always putting A at the first location across all the positions in the array. But what if instead of having A, C, T, G as your alphabet, you have probability vectors? So maybe with 25% probability it's A, with 50% probability C, and so on. So that dramatically increases the alphabet size, so you don't have to synthesize as long strands as you would need as with a quaternary alphabet. So that's amazing. So of course that presents new challenges as to how to design error correcting codes in this framework. Frederick is working on that. You can ask him more if you want. Yeah, and another thing is, of course, besides the synthesis cost, is the speed of sequencing. So Illumina is the state-of-the-art sequencing technology if you consider accuracy. So from what I've heard, is practically error-free. But nanopore sequencing permits real-time analysis, which is amazing for us because it's a bit more flexible if you want to try and integrate error-correcting codes with that. So it's also important for adaptive sampling because when you're sequencing DNA strands, you have multiple copies of the same strand, right? So you don't want to read something that you have already read before. So adaptive sampling would be great. So if you read part of the strand and you realize, oh, I've seen this already, then you can discard it. So nanopore would allow for that and Illumina wouldn't. But again, if you really want to design error correcting codes for nanopore sequencing, as I said, you need to be efficient. So you really need to tailor your codes for the nanopore sequencer. Now, the problem is that they keep changing their chemistries. 3 years ago, the width of the nanopore was 5, and today it's 9. So as coding theorists, what do we do? Right? Yeah. Another problem that's coming up right now is how do you incorporate classical cryptography notions in DNA-based data storage? So cryptography has this rich literature about how you characterize the security of a specific scheme. So usually it's defined by how many operations you need to break a cryptosystem. How does that notion translate to DNA-based data storage? We don't know, and people are still debating what metrics to use. Yeah, that was it for me.
Okay, thank you. IOBNT, I think we've seen yesterday and today that IOBNT has a lot to do with medical applications and with communication within the human body. And I just wondered when you asked the question, when I listened to the others, whether it's the first time or whether it's valid to say that it's the first time that the society maybe won't let us communication engineers do what we want. So far, I mean, we never had real problems, maybe except for my wife saying, don't keep your smartphone in the sleeping room and so on. But but did we have serious problems in implementing our technologies and in realizing applications? Well, this time we will. I'm quite sure, and for that reason, it's extremely important that much more important than so far that we get in touch with other research communities, especially with the medical and life science people. And for me, that's been the most important step in the last 5 years that we've been able to get them interested. I remember a very a meeting of our medical faculty where I was invited, and it was roughly 10 years ago, I would say, when I first started discussing the idea of nanobots being somehow active in the human body and doing something. And I presented this idea in the medical faculty. And after my talk, just 10 minutes, there was no question and no one talked to me anymore. So no one was interested and everybody thought, sure. And now this has completely changed. And I guess the reason for that is that there have been a huge number of publications in Nature and so on. So in the journals that medical people read and are interested in, and that has changed the picture a lot. And in our university, we are very much a medical university. There are many more people than 10 years ago. That was not difficult, but still there are many people now who are interested in body communication and all the stuff that we've seen so far. And I think this will be the challenge now is the most important challenge now is to really make them understand what we are doing and bring the things together and really try to implement the stuff within, let's say, bodies, maybe not the human body, but in mice or something. But this will be, I think, a very important step if we want to really create medical applications in that sense.
Okay, then I start here. Perhaps Mohammed, unfortunately you are the first. Then start with your.
So I should ask some questions.
Yeah.
So maybe from Johannes, so about the identification for the K identification for the latest channel model that you are using. Maybe can you explain the submitted version to the ISIT conference, I think, or yes, for the K.
So first of all, it's not yet submitted. It's just being worked out. Maybe first I tell the people what K identification is. So in identification, the sender and the receiver each pick a message, and the receiver wants to know, are they equal? And in K identification, the receiver instead picks K many messages and wants to test, is the sent message one of those? So so the channel model that I've been considering, or that I am considering currently, is also just a discrete memoryless channel. So for every, or as far as I can stretch it to channels like general discrete channels without any additional assumptions. So where you don't impose a particular time structure, so discrete memoryless channel, every symbol is mapped with a certain probability to another symbol independently of each other. And of course, I'm also looking at noiseless channels. Yeah, maybe something interesting for people about K identification is that with some mild assumptions, you can also decode if it's within your K set. Like if you have tested, it is one of the K, then you can know what is it. So I think K identification is kind of the very much more flexible version of identification, because you can do all sorts of things when it's in the K set.
Okay, I would have a follow-up question, but I think this is too technical, so I would rather prefer to ask something else. There was they mentioned this idea of different notion of value of information. And there is a recent trend, if you go to 6G conferences, of semantic and goal-oriented communication, where basically the pure transmission of bits is replaced by you want to induce a certain activity or a certain response of your receiver. And I was wondering if this idea of semantic communication is related to what you mentioned with respect to these other types of value of information for specific cells, or yeah.
So very, very on topic. So yes, I have to start with this. So why didn't Shannon define semantics, right? Because if you look at how semantics can be defined, there is no single way to define semantics. It's actually very ambiguous. So what is semantics, right? We say, yeah, it's the meaning, but what's the meaning for you? And it can be different from the meaning from my meaning, right? And so I know that in 6G, there is this goal-oriented and semantics, and there is a lot of work on that. But I want to argue that biological systems are a little bit different. Just in a nutshell, how a biological system, information can be seen according to a goal. So for example, there is an animal and is hungry, like for example, a lion is hungry or a tiger. I have that in my slide, that's why I have it in mind. And there is a gazelle passing by, and there is this visual information and maybe also the smell. And that information has value because the tiger knows that if it starts running after the gazelle, it can actually have a meal, right? But then if we look at different times the tiger lives in its life, that information regarding the gazelle can have different meaning. So for example, let's say that the tiger is very thirsty, and the gazelle passes by, and it can run after the gazelle, or it can go, for example, to a lake or to a pond to drink. Then the semantics there is a very lax concept, because the tiger can give a different value to the visual information. So sees the gazelle, sees the water, and is thirstier than hungrier. So it will go towards the water. And so the semantic nature there, actually you can see with this simple example that semantics is very fluid. Like, what does that mean, right? And for example, the tiger might not care about the color or the age of the gazelle. I don't know, for example, or it might not care about the color of the water because, you know, but we might care about the color of the water because a brown water maybe does inspire us to drink it, right? And so with that, I want to say, and so that's why we, coming back to the concept of semantics of information, we think that we have to look at semantics from a different point of view for biological system and look at the subjectivity of information. And that's what I will talk about tomorrow. And so we say different biological systems in different moments and for different individuals, even if they are the same species and they are in the same context, they can give different values to information according to their internal state, which is being thirsty or hungry, for example. And we start from there to define a kind of semantics that in our case applies to biological system. And it's been like 4 years we have been working on. It's an entire PhD, actually. My student did a PhD on that, and he's graduating next month. And what happened is we looked at this system. We developed a simulation environment to measure. So we got data, and then we developed a method to give a number to this subjectivity. And then we also solved it analytically. So we have an analytical info theory result where we say, this is what we think is subjective information. And it's just the tip of the iceberg. It's not definitive. But we think that we are into something there. And so when people tell me, yeah, and you ask, what is the connection to semantics? Yes, this is a part of semantics, but it's a different kind of definition that we think applies better to the biological context, to the way biological systems process and use information. There might be other semantics which are more machine-based. And so, for example, you have a digital message, right? And you have a communication system. Now, semantics there, for example, is the value of the bits that you're transmitting in a certain moment. So that is another definition of semantics, which is a little bit more probably grounded, right, with respect to how the biological system work. But yes, it's definitely connected. And I am very happy that people start thinking about semantics because I think that that's the natural evolution from Shannon theory. Yeah.
Somehow a comment from myself. So what you described with a tiger is in principle some kind of identification because the smell, so if the smell is the send messages, the send message, then of course no one knows in which the receiver is interested in. So, and therefore when the tiger is interested in something to drink, then he ignores this. And if he's interested in something to eat, then he just makes some decoding. Is it something to eat? Yes or no? And therefore it's somehow it's a little bit more related to this identification, which for some reason they put it now also under semantic communication sometimes, where I would put it more to goal-oriented communication. But I think oh, okay, yeah. Yeah.
For the audience,
probably one term, goal-oriented and semantic communication for most people.
Yeah, I think the main problem is that semantic communication, if you listen to a talk, it's always different defined. So some people have definitions, and then you are happy with these definitions, go to the next talk, and they give another definition. Yeah. But Shannon also thinks about this. He has these three things, and he wrote about semantic. He said he is on they have this levels A, B, and C is the semantic communication, and that was already Shannon wrote about that. So it's somehow he thought about this also. No, it's not well defined. No, it was not definition. It was just what kind of layers are existing. And of course, you can in mathematics, you can say that is our definition, but then another one came, and so it's not clear at the moment.
Yeah. So I had a question in direction of the IOBNT, how you envision the nano robots to be in the future. To give you some context where I'm coming from. So in end of last year, I talked to some system biologists. And for me, it now feels like they have the same vision, just that they want to engineer biological organisms to do somehow the same task, to deliver a drug somewhere specific, to do some micro operations somewhere in the body or outside of the body. Do you see this as two separate communities, or will they grow together? Do you see some synergies there? Or how do you envision this as in the future?
Yeah, good question. Actually, I'm not sure if this is already, let's say, one community where everybody knows everyone else and so on. So there's a number of different approaches. So last year I visited a medical research group in Harvard in Cambridge, and they did a lot on DNA origami and self reproducing structures and so on, which is looked to me much more complex than the stuff that I presented today, but much less complex than what you just proposed. Yeah, so artificial, biological, I don't know, robots or whatever you would call them, and then, for instance, stuff that you do and that we do, we have never talked to each other. We even didn't know each other so far. So, and this is true for the whole area of DNA. DNA storage, I think DNA storage and DNA computing, probably they also don't do very much together. At least that's my impression so far. So there are still a lot of islands which are not very well connected. But well, the more workshops like this one here happens, and the more people publish on their results, the more the people will talk to each other and bring the ideas together. So it's hard to answer whether ideas like artificially produced living bots, more or less, if this will really become reality. I don't know. I have the impression that the approaches that we have, or maybe we can also include Max, who can probably say something about spines and all that stuff, which is also an approach to implement devices. This looks to me much less complicated and more realistic at the moment, at least, without exactly knowing how far the others are. But the medical people from Harvard, they really had very interesting, very nice results. If someone wants to look this up, they have developed something which they call the crisscross architecture. Published that in Nature just recently. Very interesting for amplifying very small little biological signals. So once they detect such a signal, they are able to reproduce this the structures which signal that something has been detected. and can make it much more likely that this really will be detected in the at the outside. Yeah, so very interesting approach, and technically, obviously, also quite well feasible. So things like that seem, to me, look much more realistic to be realized at the moment, at least.
I actually also had questions in the direction of nanobots, but I think you mainly answered it, because I also was not sure, especially from the perspective of then people, if they would even want to get injected such things. But yeah, so, yeah, exactly. But maybe to change a bit the topic, I can also ask Anisha. You mentioned, I think, also in your talk a little bit the error correcting codes and the construction of such codes, and we discussed a little bit also like some ideas with convolutional codes, and what challenges do you see in that field?
Yeah, so firstly, a lot of people are not convinced about convolutional codes. Yeah, in the 60s, 70s, they were super popular, and then recently, like LDPC codes are all the rage, and people stop believing in convolutional codes a bit. But what's very nice is that it has this temporal structure, like you can describe it like a tree, and it merges in a very friendly manner with how base colors work, actually, because base colors also work in the sequential manner, because it tries to guess, okay, the first sample in the current signal maybe came from this Elmer. And then because the window is kind of shifting by one position, so the next Elmer can only be one of 4 possibilities, because only one new base is coming in. And so convolutional code, decoders for convolutional code also have these very nice temporal structures. So I think the memory of the POR and the memory of the convolutional code are really like, they can work together and give us good constructions and help us gain in redundancy. But again, the problem is that nanopore keeps changing its keeps changing its chemistry, so the base calling technology also changes. So that's a problem. And also, I would say that convolutional codes on their own are not enough. Usually, the best thing to do is have a concatenated coding scheme. So you have an inner code and an outer code. And these two coding schemes usually have complementary properties. So whatever the inner code can't deal with, maybe the outer code can deal with. So it's not clear how you would go about choosing your codes and then designing or optimizing them jointly so that you get a very nice coding scheme in the end. Yeah, also I would say that because you have multiple copies, noisy copies of the same code word, let's say, how do you exploit this multi-copy gain in conjunction with how you're optimizing the inner outer codes? Yeah, still unclear.
Yeah, actually, I had more a question than a comment, but I would like to follow up on what you just said, because I mean, my background is in wireless communication, so there are some analogies between wireless communications and your work. Well, in wireless communication, people insert pilot signals to detect the status of the channel, right? So maybe could be a solution, or I don't know, to insert some known subsequences to detect the status of the nanopore, if it's doable, or something like that.
Yeah, the problem is that usually people do experiments on the nanopore and measure the characteristics of the channel, and then they fix their code books. Yeah, and then once you change the sequencing technology, maybe with the new sequencing technology, your code design isn't optimal anymore. Yeah, so once you have fixed your code book, yeah.
All right. Maybe this was already addressed somewhat in the previous questions, but I was interested in, because we have read some DNA tiling technology that's kind of working on the nanoscale, and then the DNA storage technology, which as far as I understood so far, is more like a macro scale thing, right? So I was wondering, in both fields, is there like, what's the status around thinking of storage on the nanoscale? And is there any thoughts of connecting DNA storage technologies with the nanoscale from the other side? So is it more of a macro scale technology, or is there any way of like this nanopore sequencing or something happening only on like a very small scale and then taking some information out of that? Or is it so far only a solution, or thought of as a solution for like the big storage problems, yeah, basically on the macro scale?
So from the DNA tile of view, I said this morning that the DNA computing community, they are very happy with their models, and obviously not too interested in real implementations and so on. So I would assume they don't care too much about storage. When I think about our specific medical examples, we are in the nice position. We don't need storage. We just want to detect something and communicate the fact that we detected something at the moment. But maybe there are other applications where this could be used. And I've always, I already also had this in mind, whether it makes, not only since today, not only since your talk, we discussed this in our group, whether it makes sense to look at DNA storage technology and try to combine it with what we are doing. But at least for now, we don't need it for that. So we only need computation, communication, no storage so far.
Yeah, about the macro scale storage, I think one major hurdle in DNA-based data storage right now is still random access. So we don't know, of course, sometimes you don't want to read a complete file, you only want to read parts of it. It's not completely clear how to do that yet. So some preliminary solutions that were suggested is that you have like a huge pool of DNA strands, and you tag local regions with specific primer pairs. And when you want to recover a part of the file, you only amplify using the primer pair of the corresponding region. But then, as you can imagine, it's not a super scalable solution. So yeah, people are still, we are open to ideas.
Okay. So maybe it's a jump. Yeah, it's a different topic, but you know, in conventional communication, patterns are pretty important, especially if we are talking about applications in industry and so on. What do you think is the role of patterns in the IOBNT or molecular communication framework?
Okay, I was hoping not. No, just kidding. So, okay, this is a very important question, and it's probably the question that every one of us asks themselves, because so patents are designed to protect ideas. In my view, they might be also detrimental to development. Meaning that when we are after a patent, we kind of want to protect our own turf and we want to make money out of it. While, for example, in forums like this, we are just sharing ideas and we don't care, right? We just want to develop and we don't, I mean, for example, my position is always, I hear what others are doing and I am so happy, although I'm not participating, well, I am participating here, but I'm not benefiting from all these German movement and funding. I'm so happy to see this development because I think the whole community benefits from the great research that you're doing here. You are advancing the world. And the same when I see somebody else who takes molecular communication to the next level, like all of you are doing. I'm just so happy. But what if we had patented the first ideas of molecular communication and then siloed, then we would have stopped all this development. And so when is the time to have patents and say, I want to make a commercial application? I think that there are many ways in which molecular communication can become commercial. And at that point, it's a fine line, right? So for example, in my university, we have an office where you go, and I guess in most of the universities, we can go and tell our ideas and say, do you think we should patent it? And so there is an officer who looks, and there is a committee, they look into the idea, they look into what is patentable, and then they can propose. And then if that becomes true, then we have the basis for a startup company, right? And so some of my colleagues went down that road. Some of them were unsuccessful, some others were successful. And it's not a measure of how good they are. Sometimes it's just luck, right? And you just hit a subject that becomes a very requested commercial product. Some of my colleagues lost track of research because of that. I don't know if you view it as sad or not. They are very happy. They don't do research anymore. They took a leave from the university and they are 100% on the company. They are happy. They are making tons of money. They can even, go away without their normal salary as faculty because they make enough money, but their time is 100% on the company. They are happy, but they don't do really research, like academic research anymore. They almost not publish papers, but they are doing something with their life, which is very valuable. So I think it depends on what we want to do and who we want to be. Yeah, so that's my take.
Yeah. And I think it also depends on how you are trained. I guess many of the guys here in the room who are electrical or communication engineers are much more likely to think in terms of patterns than, for instance, people like me who are from software. So from the beginning, typically you learn at the university as a student or whenever you talk to companies and whatever, that it's very hard to patent software, at least in Europe. Typically you don't even think about that. Yeah, and I think this really has to change. We had to, Shashwati, you might remember, last year at the workshop in Berlin, when we talked about our latest ideas in this and that, and then there was this lady from Charité who afterwards, over a coffee, came to us and said, "Are you crazy to talk about that? You first have to patent that before you..."
Yes, exactly.
Yeah. Yeah, exactly. Yeah, exactly. So I myself, I have one patent together with someone which is actually it's Shandor Fekete. Eduard, you will know him. He's a faculty member here at the university. In 1.5 hours, he will sit with me in a Vietnamese restaurant and have dinner, and we party over our patent. No, but the patent is already something like 15 years old, and no one has ever asked to license it, so it's not really relevant. But it was a nice experience.
So perhaps a final question, since we are almost done in time. Someone have an urgent question or something like this? Oh, okay.
I don't know, maybe this is a stupid question, but yes, it's in my mind. So when we are doing this channel modeling or this molecular communication work, and we are in our results, we show, okay, this is the bit error rate, this is the SNR value, and this is doing this thing. Okay, the bit error rate is good or the SNR is good. But then how do you think it will benefit the life science or the medical applications which we target? Like in the long run, when we're talking about molecular communication or IOBNT, we're talking about early detection or localization or targeted drug therapy. So what is your suggestion on this? How would this output or metrics which we evaluate would like affect the application scenario or in the long run?
I don't know who want to answer.
Yeah. So I think this also touches what you said with this semantic information measure, because I think, like, of course, you can look at all kinds of measures to quantify something that's going on in the communication, or to quantify something. And I think usually this something is some sort of efficiency. So I think if we have the performance measures for what we're doing that are tailored towards the application, like the medical treatment, then you can quantify somehow the efficiency of the medical treatment, maybe, or some complexity of it. And information is also just like a way to describe complexity. So I think it's I think when you find the measures that quantify the performance that is relevant to you, then the measures are valuable. And of course, first you try out all sorts of measures on the way to find the properties of the measures that you have now for the new applications, or with respect to the new applications, and then also to find maybe measures which are better for that application.
If I can add, so we were talking over dinner with Max there, the other Max, and he told me something that actually stuck with me. That is, when we have a research project with the life science, we have to give back something to them, right? And you said you cannot only take. That's so true. We have to give back something, and that giving back is finding the application of what we do in their domain and how can they benefit in their domain. It can be a practical application, right? For example, I'm working on drug development now. But it can be also an application to their discipline in general, like discovery of a process, or something that benefits them educationally, like educational tool, for example. I have a very good friend. He's a faculty in my university in biochemistry, and one of his major breakthrough was to build a simulation environment that now is using biochemistry as an educational tool. And he got tons of money for that development, and he became famous as the developer of that educational tool. I don't know if you ever heard about the Cell Collective, but if you Google it, you will find out Tom Elliker, and he's a very good friend of mine. Like our families are friends, and so that's one example. I mean, he's not in molecular communication, but that's one example. He's a computer scientist / biochemist, but he gave back to the biochemistry. He doesn't have a wet lab, so he does only computational work. He's a bioinformatician. But he gave back to biochemistry, just saying, look, I developed this simulator. Now you can use it in your classes to teach biochemistry, and students were able to understand so much better. That's a success, a true success in research, and you gave back to that community. So yeah, thank you, Max, for this. The other Max.
Okay, then perhaps a final question of my side for everyone here in the panel. So if I give you 1 million euro now for a small project for 3 years, what would be your goal of an interdisciplinary research you have in your mind for the next 3 years in molecular communication together with your research, perhaps?
Okay. I don't know about the million, but let's say if I have money and can choose something interdisciplinary, I think to connect it to my research, I think it would be somehow about modeling. So I think when we're talking about this goal-oriented communication, about different measures for semantic information, or so about new applications, and especially I think with nano computers, about low complexity things, I think there is kind of a zoo of possible goals. And I think in the passive information theory, if I'm correct, it wasn't that big as it is now. So maybe the channels varied a lot, but the problems didn't vary so much as they did now, maybe. And also, I think when you have more communication between machines, between nanomachines, or between computers or something, somehow the goals, the communication goals are much more, or you have to be much more precise on goals than, for example, if there is a human who does error correction afterwards. So I think for me, it could be at least about somehow goal modeling with goal modeling and developing a theory for classes of goals. Yeah. And probably trying to find some goals for which you can be super efficiently.
I talk too much, so.
Well, I'm not sure in my case if I also did do that, but anyway, very simple proof of concept. We would realize this the DNA tiling stuff for a medical application in oncology, and we have people with whom we can do that together, and it would be together with Shashwati and her group, and Charité and our UKSH, that's the clinic in Lübeck and Kiel. I think this looks like a promising and maybe successful combination.
All right, so right now I think most of the focus on DNA-based data storage is to store DNA in this powdery form. And in that scenario, of course, you still have a billion types of errors. But I think it's more interesting, of course, more challenging to try and store information in living organisms because like it could self-reproduce and stuff. But of course, again, I don't know what kind of errors happen with what frequency. So it would be cool to work with biologists and to identify which kinds of errors are more dominant. And then not just try and design an error correcting code for a specific type of error for this many errors, but like having something all encompassing in this model.
I can close. So I think I wish I could develop preliminary results in some information modeling, and I mentioned the human brain. And so I already have an ongoing collaboration with the neuroscientists, but that million I would spend in getting very good data for the modeling. I think that's the way I would spend that. So, and data is very expensive, and I would share it with the community, of course, but I would do that. Very, very good data.
Okay, then I would say thank you all, especially the people in the panel, for these nice ideas and the nice answering and interesting opinions. So thank you.